screen. Yeah, thanks so much, Hafiz. Yeah, it's so fun to be able to be here. You can see this, right? Yeah, it's funny, I usually start these talks off by mentioning my trajectory, and actually I often credit Guelph. I'm not lying, I do that. Yeah, it's kind of neat because, I'm a community ecologist. I was trained as a community ecologist in Ben Gilbert's lab, but of course I have this history of being in this department in Guelph, which in Hafiz and Chris's labs that are very strong in evolutionary ecology. And I feel that kind of like set me up for a lot of the things that I'm currently doing and thinking about, which is also kind of reinforced the University of Toronto, which is very much like population genetics, like hardcore evolutionary biology, and kind of being in that environment as an ecologist. But I'm actually going to start off by reminding you of some issues that were going on in ecology. In the past 2 decades, there were calls questioning the validity of community ecology as a science. That was based on criticisms that communities were just too complex. They'd never be able to achieve the mechanistic power of population ecology or the broad generality of macroecology, or things that span broad biogeographic gradients. But I like to argue that populations are embedded within communities, and communities are embedded within these broader landscapes. And so the community really is this like critical characteristic scale in order to understand what's happening at all other levels of biological organization. And so before I get into the specifics, I'm going to tell you about the study system that I mostly work with. This are annual plant communities for Mediterranean climates back in California. I really like working in these systems because they're biodiversity hotspots, they're hyper diverse. Annual plants are amazingly amenable to experiments. You can just move them around as seeds, put them anywhere on a landscape, they'll mostly stay there, and actually measure all components of fitness in a single growing season. And that can happen in the greenhouse, it can happen in the field. And my approach to community ecology is to kind of combine in some ways population ecology and community ecology by, doing kind of tightly controlled experiments based on fitness, where we might ask, okay, how are these different species performing along environmental gradients? What are each of them doing separately and why? And what happens when we kind of stack them together to form a community? And so yeah, really combining this power of population ecology with community ecology, and in different ways, kind of combining scale into this to kind of get a bit more at the biogeography and the generality of things. And one thing I want to point out too is that, because of the pandemic, we've had a abrupt switch in a good way. I think this is a silver lining of the pandemic, because we haven't been able to go to California in the past 2 to 3 years. Which had a whole bunch of funny consequences. We had to rescue experiments and all kinds of stuff that were kind of trapped down there. We've switched to working with flower beetles, which has actually been a lot of fun. There's a lot of benefits to working with these mesocosm experimental systems, doing experimental evolution and all kinds of things with them. So you won't see any flower beetles in this talk, but I bet you if I were to give a talk next year, it would be like 80% flower beetles based on what we have going on right now. So my lab, generally, nothing's off limits. We do a lot of projects on, species coexistence, community assembly, metapopulation dynamics, local adaptation, character displacement, and even speciation. I think the one thing that combines all of these themes together is a focus on population growth, a focus on population persistence, and just parameterizing models that can estimate things like how intensely species are interacting and how they perform in different environments. If you look right here in this one quadrant, I don't know if you can see my little cursor there, but that's Tess Granger, who's currently, she's an independent postdoc actually at UBC, but she's been working with us with the flower beetles. So there's her with a whole bunch of little beetle mesocosms. And of course, she'll be starting as a faculty member with you sometime next year. It's exciting. So I really wanted to point out a couple specific projects that I thought, go into those in more depth, that I thought might appeal to a number of people here. I'm kind of loosely organizing this talk around some themes that are kind of more on the community ecology, kind of fundamentals side of things, and then ending with more on the evolutionary ecology side. So that's where we're going to start. I want to start with something that's probably familiar to a lot of you, especially if you've taken an ecology course or taught an ecology course, and that's this idea of the filtering metaphor in ecology. Usually when we're trying to teach students about the community assembly process, we show them a cartoon like this. With this cartoon, it's saying, okay, there's some kind of regional pool of species that have some traits, which I've just denoted here with their colors. And we're trying to understand, like, how do we get from that broad regional species pool to the specific species that live in any one local community, right? And the schematic that we like to show is kind of this regional filter kind of passing through these kinds of filters that select species out that have different traits. And so we think, okay, maybe first the environment is happening at a larger scale and it's selecting out species with particular kinds of traits. So here they all have a blue phenotype. They're all blue and suitable for that blue environment. And then competition is selecting maybe for differences among those individuals and those traits. And so here we can see, even though they're all blue, they're as differently blue as they can be. And those are the ones that were able to persist in that local community. And so this is how we teach things. And when we see patterns like this, we think, okay, this is evidence that there's really strong environmental filtering coupled with some competition. We see these trait patterns distributions. Of course, there's some issues with this, and the main one that I'd like to focus on is that environmental filtering is not likely independent of competition. So if we think about environmental filtering in the strictest sense, it's basically saying that like this species would be unable to live in that environment regardless of all kind of other things, sort of it's outside of its fundamental niche. And so if it were to be able to get there, it would be unable to persist. And so you find it in some places and not others, and that's because of the environment. Of course, we know that there are other processes in ecology that can lead to essentially the same pattern. And so here's a case where, we have the species that in the absence of competition with another species, it might be able to live in both of these two environments. And it isn't until this other dominant competitor in that white environment is there to actually exclude it, and essentially leading to this exact same pattern. And the trick is, in nature, there are other species, there are other interactions, there's competition. And so I would argue that the competition scenario is kind of more realistically what's actually happening in nature compared to the more unrealistic environmental filtering scenario. The trick here is there's this kind of survey meta-analysis from 2015 by Nathan Kraft, which is a paper I really like to mention because this is probably I think that's the second paper I ever peer-reviewed. So it was many years ago when it first came out, which is kind of fun. But they found that 85% of studies tend to confound competition with environmental filtering. And so I have a question, one question for this is, how do competitors influence evidence of environmental filtering? How are they kind of working together? Like, how does competition kind of change the story here? And so what does it mean to be filtered out of an environment? Basically, what we're saying is that if these species were kind of placed in a local environment that was bad for them, they should actually fail to persist, right? And that would be what we consider evidence of environmental filtering. So how do we measure persistence? The way that I like to approach questions, and you'll see this as a theme through a number of different projects I'll be talking about, is to measure things that have to do more directly with persistence, like population growth rate, or specifically invasion growth rates is what we call this. And what I do in this case is I'll pick out a pair of species that compete. Here we've got just a cartoon, Solicinia and Lupinus. And what I'll do is I'll grow each of those species alone in the absence of the other species at a low density, so just essentially just experiencing the environment. And then I'll grow them at low densities within a background of the other species, specifically at the other species, single species equilibrium. And from that, I'll measure their population growth rates. And here I've denoted this as the population growth rate, or lambda, when they're just experiencing the environment in the absence of other species, versus their lambda when they are experiencing that other species. So you can kind of see we're adding on the competitor environment on top of the abiotic environment. We essentially ask, is this population growth rate below 1, indicating a failure to persist, or is it above 1, indicating that they can't persist? I replicate this, in this case, in 2 soil moisture environments, a wet and a dry environment, and replicating this across 30 different species pairs. So I'm going to walk you through the results here. The take home, as you can see, is that environmental filtering only emerges with competition. I'm going to show you first a case where we don't have competitors. And just to walk you through this graph, we have the 2 soil moisture environments on the x-axis, the wet and the dry. We have our invasion growth rates, again, in the absence of competitors on the y-axis. And here, this little line is just denoting the kind of boundary between persistence and failure. So you want to be above that line to persist. So I'm going to throw a bunch of points on here. Any point that's connected by a line, that's actually the same species pair in those 2 different environments. You can kind of see how they kind of jointly do as we can force them to be in a dry or wet environment. And in this case, what you can see is most of them are above this line. There's very few species pairs that are actually unable to persist in one of those environments. And so, yeah, overall, not surprisingly, it's a greenhouse environment. It's suitable and it's within their environment. We can contrast this to what happens without competitors. So again, the same species pairs here. And now you can see everything's sort of shifted kind of dramatically downwards. And if we were to compare this, we would see that, yeah, a whole bunch of species are filtered out or fail to persist in those environments. It's actually fairly equal across those two kinds of environments. And we see a lot of cases now where they cross that line. So like a species might be able to persist in a wet environment, but not a dry environment, for example. And so what's happening here, this is really just indicating that in this case, competition is what is generating a strong signal of environmental filtering, right? And so if you just kind of measured this in the field, you might conclude that, okay, it's all just the abiotic environment, whereas this is really entirely driven by competitors. I think it helps to really focus in on a species pair to just kind of see this in a different way. So here we're dealing with two Licinia species, Californica and glabrata. If we first, again, look at their population growth rates in the absence of competitors, you see they both do very well. They both don't really care about the soil moisture environment. They're both kind of just chilling at the top there. And they're kind of equally good in those two kinds of environments. It isn't until you contrast this again with competition that now you see everything's downward shifted, and you see actually opposite responses where, glabrata prefers a wet environment and Californica prefers a dry environment. There's actually something extra you can see in these plots by just kind of looking at any one of those species in that pair. And so let's do that. We can actually compare the population growth rate in the absence of competition to the growth rate with competition to tell us how strong competition is in those two abiotic environments. And so I'm just denoting that with an arrow here. So the larger that arrow is, the stronger competition is. And what you can see is that competition is just stronger for this one species in one environment, the dry environment, compared to a wet environment. And again, I think what this is showing is that the environment is having this reciprocal effect now, where it's actually impacting the strength of competition. So it's very much not just like competition is affecting the environment, but also reciprocally, the environment is also affecting the strength of competition. Now, because of this, if we look at how these species kind of jointly do in those two kinds of environments, what you can see is that because there's these kind of opposite responses and how they respond to a wet and a dry environment, both of them actually fall above that persistence threshold in a dry environment, whereas in the wet environment, it's the glabrata that's competitively dominant and should exclude the Californica. And so, again, the environment and competition are reciprocally intertwined, and this is enough to actually affect not only if those species persist individually, but also if they coexist together, where you expect coexistence in a dry environment, but not a wet environment. And so that leads to this question that was posed in a tree paper by Carolyn Tucker, Mark Cadott, and I believe a third author, I'm not remembering. But they posed this question of, should environmental filtering as concepts be abandoned? And I'd say a response based on this data is that on one hand, yes, most environments contain competitors, and competitors strongly modify persistence. But on the other hand, no, maybe we're asking the wrong question. I think we've been able to reveal this kind of interesting interplay between species realized and fundamental niches. And if we're thinking about why did we even come up with this filtering metaphor, probably it's something to do with simplifying the problem, maybe clarifying teaching a little bit. But I'll say I've started teaching, my second year ecology class both ways. I've used the filtering metaphor first, saw the confusion on their faces as soon as they started to ask probing questions about the concept. You'd have to kind of tell them oh, it's just a cartoon example. There's a lot of cases where this metaphor kind of falls apart. And then all of a sudden the undergrads, they don't buy ecology as a field anymore, right? So I've been teaching it instead from a perspective of these kind of 5 core ecological processes that each have their own specific influence, but also can interact in different ways to explain all kinds of things, the population level, the community level, the ecosystem level, etc. And I found that even though this seems more complex, it's actually made and streamlined teaching in at least my class. So yeah, sometimes simplicity could actually lead to some issues. So in this project, I was very much looking at the interaction between the biotic environment and the abiotic environment. I've done a bunch of other projects to incorporate kind of other kinds of pairwise interactions, the biotic environment dispersal, things like that. What I will say is, most of our community assembly models involve all of these 3 things, and not only the pairwise interactions between them, but how all 3 of them are kind of operating at once, right? But you can imagine this is very difficult to test. How do you manipulate all three of those things, right? And especially, how do you do this in nature? I've just showed you. And to do that, I want to examine this idea of what mean individual fitness is and compare that to per capita population growth rates. These are two kind of entities that are probably familiar to a lot of you. If we think about mean individual fitness, that's something we think about as being important in the realm of evolutionary biology, say local adaptation studies and things like that. Whereas per capita population growth is more in population ecology, right? And it's really, you can kind of look at the feedbacks between these two things as something that people have looked at to kind of look at this interaction between evolutionary ecology and community ecology. But one thing I think is really interesting is that it might not be immediately intuitive, but these are literally exactly the same thing. Mean individual fitness is per capita population growth rates. And so we can actually look at the distribution of individuals to understand what's happening in populations. And so if we were to do this, what I'm going to plot here is a case where we have the individual fitness here averaged for different populations at a 10-kilometer scale, right? And so you can see some populations on average are in a better environment. They're doing better overall. The points are higher. And there's some distribution where there's some rare ones that are doing well and some many populations that are doing quite poorly. But we don't often think about what's happening within each of those populations, right? Again, the mental model that we might intuitively have is that, okay, if the population growth rate is 2 on average, we might think that all individuals are contributing 2 offspring to the next generation, right? We just got that average and it's saying there's 2 seeds per individual. That's the per capita population growth rate. So everyone's contributing and everyone's contributing kind of equally. If that were true, if we were to take all of the 10 individuals that are within these populations and we kind of ranked them by their fitness, kind of like a rank abundance curve, but in this case it's for fitness of individuals, it'd be kind of a flat line, right? That's indicating that everyone's contributing. Instead, if we look at the data, it's actually something more like this. And you can see a bunch of things here. The first is that these populations are essentially being led by a few exceptional individuals. You see this huge unevenness in how individuals are contributing to those population growth rates. In some cases, like this single individual is responsible for the population growth rate of that entire population, right? It's got like 100 seeds on average. And so this kind of mental model we might have that everyone is contributing is probably not true. I'd like to say this is an exaggerated example, but I don't know. I mean, serpentine is kind of a weird environment, but again, I'm sure if people did these transplant experiments elsewhere, they'd kind of find something similar. Whereas instead, we find the populations are really being carried by a few exceptional individuals. So more in this case, where you have 20 and 0 and 0 and 0. And so relating that to the data that I just showed you, basically what I think is happening is we increase the spatial scale of our population kind of averaging. There's a much higher chance that we're including these exceptional individuals. And when we do, it does 2 things. It drives up the population growth rates, but I think it can also feed those sink populations where those 20 seeds might fall into adjacent sites that are actually unsuitable and actually kind of be the source that creates those sink populations. And so, yeah, I mean, what I just showed you is a case where we were actually able to look at these kind of main kind of core processes that underlie community assembly and do it in a natural landscape. And actually, all the data I just showed you is from the abiotic environment where we have removed those competitors just as a first pass. But again, yeah, this is a data set we just started working with, and there's all kinds of things that we're doing with it right now beyond this, but I wanted to show it to you because I'm very excited about it. So I thought we'd switch now to thinking more, it's kind of similar ideas, whereas community assembly processes, there's feedbacks between the biotic and abiotic environment, but now trying to understand this through the lens of evolution. And often if we think that species are too similar or too competitively asymmetric, they should be unable to coexist with each other, right? And so you can see these 2 species here that we're kind of using their roots to show their niches, and they're similar. They're accessing resources at similar depths in the soil profile. So if they're too similar, they basically have 2 outcomes. One is exclusion, either the random exclusion of one of them if they're interacting neutrally, or the exclusion of the inferior competitor. But they have the second option, which is evolution, and that's the evolution of differences that actually can allow them to persist and coexist together. This basic process of evolving differences that might help them coexist is underlain by this process we term ecological character displacement, which is the process by which interspecific competition drives evolutionary change. Typically, when people are examining character displacement, this is the pattern that they look for. Like all the studies on character displacement look for this, where you have two species that occur in symmetry or together in the landscape versus an allopatry, where species one is living alone and species two is living alone. And we usually look for patterns of phenotypic differences. And so the expectation is that in sympatry, where they've had that history of interacting and selection to kind of drive them apart, that you would expect traits to be greater in sympatry, which you see with these arrows, compared to if you were to look at those traits in allopatry. So you see in allopatry, species one and species two are pretty similar. Character displacement is an important process for population divergence, which can be used as an explanation for things like speciation, or even on broader macroevolutionary scales, some of the kind of big differences we see among species in their traits as they try to use and partition different resources, right? But what you'll notice is that in all of these examples, the focus is on phenotypic traits, and that's kind of the history of the field of character displacement. But as I thought about this more and more, I think there's a lot of cases where phenotypic traits might actually limit our ecological and our evolutionary inferences. And I just outlined a couple of examples of that. The first is that we have no ability to actually assess the ecological consequences of that phenotypic change. We might know that two birds have different beak lengths, but we have no idea if that affects how intensely they compete interspecifically. We have no idea if that affects their persistence to the coexistence. We're actually assuming that interspecific trait differences actually confer a niche difference. So the more different they are, the better those two species are at living together, the more likely they are to coexist. A lot of cases where trait differences might be bad, right? So there might be a bird that's just like larger than the other bird, and because of that, it's competitively dominant. And so in that case, differences are actually a negative thing. And the last one is that we're very prone to missing instances of character displacement that might be present through unmeasured traits or multivariate trait combinations. I particularly feel this way as somebody who studies plants, because I feel like I'll never know what a plant is experiencing. I'll never be able to correctly measure the traits that are important for them. I might get leaf size or root depth or whatnot, but I'll never know like the uptake rates of specific essential nutrients, right? Very hard to do that. And because of this, not actually these specific issues, but some other ones as well, there's a tree paper by Yoel Stewart and Jonathan Lossos who said that maybe the rise in esteem of character displacement has outpaced available data, and that more complete rather than simply more case studies are needed. So I'll tell you the way that I've been addressing character displacement, kind of using similar methods to the ones we just talked about, to answer these questions of, do we see evidence of character displacement? And if we do, does it act towards helping rescue declining populations from extinction, promoting their coexistence? As you might have guessed, the way that I've handled this is to actually do competition experiments, where here I'm working with two species of grasses. We have Vulpia, which is a native species to California. It's competitively inferior to Bromus, which is a non-native competitor. I have instances where these species occur in the same part of the landscape in sympatry and have had that history of interaction versus cases where they're allopatric. That's kind of the blue and the red here. To actually assess evidence of character displacement, again, I use population growth rates, which I showed here on the y-axis, and we're specifically looking at Vulpia, so the inferior competitor. And we can actually assess this at a gradient of intra-specific to inter-specific competition. The first thing I want to point out is the more dramatically negative this line is, the stronger interspecific is relative to intra-specific competition. And what we're specifically looking for here is a contrast between those sympatric and allopatric populations. If there's no character displacement, we'd expect these lines to be parallel. Again, it's negative because Vulpia is inferior, we know that, and so inter will always be stronger than intra, but no difference in the intercepts here, or the slopes, sorry. If there is character displacement that helps Vulpia contend with competition from this interspecific competitor, so in case we'd expect the slope to be shallower for those sympatric ones that have had that history of interaction compared to the allopatric ones. That's kind of what we're looking for as evidence of character displacement impacting competition. And of course, this is replicated. I have 8 contrasts in the sympatric group and 8 in the allopatric group, which doesn't sound like a lot, but anyone who's done these experiments knows that every single data point that I'll show you is underlain by like 100 other data points that are actually used to kind of parameterize different aspects of the model. I like this method because the strengths are that it can actually detect character displacement without any actual knowledge of the actual characters. And it can actually directly elucidate the ecological consequences because we're measuring the intensity of competition, which is relevant for persistence as opposed to phenotypic traits. So the take-home message I'm going to show you is that character displacement does help rescue populations from competitive exclusion. This plot is set up exactly like the predictions that I just showed you, but just to remind you of what that is. If character displacement is providing that kind of help, we'd expect the slope to be shallower for the sympatric ones compared to the allopatric ones. And that's exactly what we see here. You see it's kind of bumped it up. And this might not look like a huge difference if you're looking at the y-axis, but it's on a log scale. So it's actually a 5 times increase in population growth rate for the sympatric ones compared to the allopatric ones. And if you're really looking, we can compare that to the persistence threshold, which doesn't actually tell us coexistence outcomes in this case, but kind of gives us an idea of like the biological relevance of this effect. And you can see that when they're mostly dealing with interspecific competitors, the allopatric vulpia just fall below that line, whereas the sympatric ones are actually pushed above it. You might ask, what's the mechanism? Is it increased niche differentiation as we think it might be? I don't have time to show you all the data for this, but definitely check out the data, or check out the paper. But the answer is no. It's actually a reduction in competitive asymmetry. So they're actually becoming more similar to each other in a way that actually benefits vulpia. So a very different mechanism than the ones proposed by the original character displacement papers. What I think is happening here, like why does this outcome differ from what we typically expect? If we think about the mental model for character displacement, it's the idea that we have 2 species that are interacting pretty symmetrically and they're very similar. And before coevolution, they'd overlap a lot in their resource use. And because of this the symmetric interactions, this should actually kind of push them apart to utilize different niches. And so that's where you kind of expect differences to evolve that are beneficial for both species and allow that niche differentiation. Again, in this case, I said we're dealing with a very different situation where one of these species is very inferior compared to the other species. And so you can imagine there's this very strong asymmetric competition where the inferior species is experiencing a lot of pressure to better contend with this bromus. Whereas this bromus isn't experiencing as much pressure from the inferior non-native species. I think this is a case where you do get this convergence of resource use because of that asymmetry that does help kind of balance them out. I really like this example. If we think about evolutionary dynamics, it's this idea that we have a change in the ecology and how species are using resources because of evolution, directly due to those pre-existing differences that those species had in the ecology. So how asymmetric the interactions were prior to coevolution. Yeah, so to me, this is like a proper eco-evolutionary dynamic because the same kind of variable is evolving and then affecting future evolution. I then like to ask, just, to satisfy, what I think is happening with traits, do we actually see evidence of character displacement if we measure these phenotypic traits? So I measured all the things that I thought might be important for these grasses when they're interacting. And I compared what those traits were in sympatry versus allopatry. I'm not going to show you the actual data because it's incredibly boring. It's in the paper. It's basically just nothing's happening for any of those traits. And so to me, this is really reinforcing this idea that these phenotypic traits that we measure might actually mislead inferences about the presence of character displacement that we can detect through a competition experiment. To me, it's kind of shocking that these 2 species kind of were able to have this kind of effect on each other, given that they're embedded within these much more diverse communities. I will say, though, that bromus and vulpia are some of the most abundant species there, absolutely. If you do like a rank abundance of these species, they're usually just like at the top. But still, it kind of highlights to me that even in these communities where there's lots of species, they can still have this kind of strong effect on each other. The last thing that I want to talk about is directly feeding off of this. But, if we think about something like coexistence or population growth rate, something like this, competitive ability, These kind of like things that we measure are really just they're estimates that are underlain by a combination of specific demographic rates. And specifically, if we were to kind of break this down, we say that coexistence, population growth rate, all these things is kind of the outcome of how species are interacting with each other interspecifically. So like these arrows here, how those species are interacting with themselves intraspecifically, and also how well they perform at low densities. So this is their low density intrinsic growth rate. And I think there's a lot to be learned by actually breaking down coexistence and seeing how these individual demographic rates evolve. One of the reasons why I feel that way is I think it provides this really nice link between coexistence and life history theory and also evolution, telling us something like the how, which is the life history, the what is like, can these species actually coexist, and why has evolution allowed that to happen? And also kind of linking scales, like what are individual traits for individuals in the population, kind of allowing population persistence that then allows the community to either be diverse or not. And what I'm about to show you is some work by my PhD student, Jawad Sakarchi. When I say that coexistence and population growth rates are kind of underlain by these kind of different demographic parameters, I mean that very precisely. And so, there's now some mathematical formulations of competitive ability that I've shown here. So competitive ability is kind of like this K of Vulpia, so KV. And you'll notice that there isn't just one parameter in here, right? It isn't all just about inter-specific competition, which might be a little bit weird. You think like, okay, if I'm trying to understand how species interact and their competitive ability, like you think you would only care about that, right? But there's actually two other parameters in here. There's the lambda and there's also the intra-specific interaction coefficient. I want to kind of explain why these kind of other two parameters are important for competitive ability using an analogy of an army. So let's picture some soldiers. So we have these two soldiers. We can think about the inter-specific interaction coefficient as defining the average strength of a soldier. So this larger soldier has a higher inter-specific interaction than the smaller soldier, so it's a better soldier overall. But the other two parameters together, the low density growth rate and the intra-specific interactions, actually define the size of the army because they're kind of involved in carrying capacity. And so this might be what this looks like. And so you can imagine that there's actually two ways to win the war. You can have really strong soldiers, or you can have a really large army, right? And so that's why all three of these things are kind of involved in a competitive ability in determining who wins competition. So for Jawad's thesis, he was trying to ask, like, how have these different demographic rates evolved in sympatry versus allopatry? I'll start with inter-specific competition because it's the one we'd expect to do something. And again, if Vulpia is evolving to better contend with Bromus, we'd expect it to evolve to be less impacted by the Bromus. And so the sympatric ones would have a smaller coefficient than the allopatric ones. And that is indeed the case. This is a statistically significant difference. Kind of a weird one is what happens intra-specifically. So why would inter-specific competition ever drive a difference in intra-specific competition? If it's actually evolving in a way that kind of benefits Vulpia's ability to establish that large army against Bromus, then this is what you'd expect to happen, is that the intra-specific competition be lower in sympatry than allopatry, which is indeed the case. And similarly with the intrinsic growth rate, how would we expect that to differ? If, again, it's trying to establish this large army, then we expect that rate to be higher in sympatry compared to allopatry. And that's one case where there was no difference between these two different groups. So why I think this is cool is we find in sympatry, Vulpia evolves to be less sensitive to inter-specific competition, with which we expect and people try to measure. But we also show that intra-specific competition is something that's evolving, and this is something that hasn't really been demonstrated before. If we kind of look at how all of these combine together to predict competitive ability, we can see there's actually a 2 times increase in that competitive ability with all of these parameters kind of evolving as they do together. So it's a sizable effect here. And there's this kind of side note where life history theory tells us that there should actually be trade-offs between these different parameters. Yet when we actually look for those in the paper, we don't find any. And in fact, we find evidence of trade-ups where things kind of evolve mutually in a way that's kind of beneficial. And we don't yet see any of those constraints being imposed. So this paper, it's available on early view. Jawad made some promotional materials, movie style. This is one of the ones he put together. But yeah, you can find it in the Just Accepted tab on AmNet if you're interested. There's a lot of stuff in this paper. This is just kind of like this is like part 1 of 3. It's a kind of a fun paper. And yeah, it's currently in the unformatted manuscript version. We're still waiting for proofs, but you can access it if you want to. So just to summarize some take-home messages from everything I just told you. The first is that I've shown you that processes do not operate hierarchically, but rather they reciprocally affect each other. It goes both ways. The environment affects competition and competition affects the environment. In nature, I've shown you that individuals overwhelmingly occur in unsuitable environments, which is just something I find kind of cool, and isn't something you could detect without doing these kind of invasibility trials in situ in the field. Shown you that character displacement acts towards rescuing populations from extinction in ways that promote their coexistence. And lastly, I've shown you that there's several ways to win competition here, including by reducing your intra-specific competition, which is kind of unintuitive, with an absence of trade-offs, which again, I think is kind of an interesting finding here. And I've kind of showed you these different aspects of scales and individuals and ecological processes and linking that to evolution, but I really do want to put these together in a more explicit way. And what I mean by that is, again, I've done some projects, including some of the ones that I showed you, but some other ones where I look at scale and how that affects ecological processes and how that in turn affects different kinds of patterns like species richness, species area relationships, things like that. It's been worked by some other people that have showed how scale is important for things like adaptation. And really, I kind of want to put all of these gaps together where we're looking at how scale is affecting these ecological processes to generate different patterns of who's interacting with who and on what time scales, and see how that affects the process of adaptation, especially in communities. But really, I started with this kind of general approach of trying to use population ecology, multi-species population ecology to better resolve what's happening in communities. And absolutely, I'm still doing this, but as I kind of look more and more into this, I find that I'm increasingly fascinated by the ecology of individuals, how individuals are using resources, those distributions of fitnesses among individuals and populations, and why that might be happening. And so I think that's going to be a major theme for me going forward, probably with the flower beetles. And so that's it. Yeah, thank you so much for inviting me to come talk to you. Really looking forward to chatting more with a bunch of people I haven't seen in a long time, so that's great. And I do want to put a quick plug that we could be recruiting for fall 2023. If you know anybody, if you want to let them know about this for graduate students. My lab I love my lab. They're so fun. I love everyone in it. Everyone gets along really well. Everyone's doing really cool research. Yeah, just, we have lab meetings in trees sometimes. This is Jawad for Halloween. He went as the scariest thing he could think of, which was Loca Volterra. And yeah, it's just a great environment. The BRC is amazing too for being a scientist. So absolutely hit me up if you're interested in that or know somebody who might be. So I'll stop there and happy to take questions.